Hello, this is Dr. Richard Louie, Center Fellow at the University of California, Davis, Lawrence Livermore National Laboratory Point-of-Care Technology Center. This presentation is on multiplex nucleic acid recognition for bacterial and fungal pathogen detection versus culture methods and the lessons learned. The learning objectives are to be able to define septicemia, describe the pathophysiology of sepsis, and to understand the epidemiology and clinical significance of bloodstream infections. To learn about current culture-based and new molecular-based methods for diagnosing bloodstream infections and to understand their limitations. To evaluate the clinical performance of a multiplex PCR method for pathogen detection and its clinical utility, and to be able to summarize the lessons learned from our clinical experience with a multiplex PCR method for pathogen detection. This is our course outline. The program is divided into 4 sections. First, Introduction to Sepsis. Second, Current Diagnostic Methods and New Molecular Tools for Pathogen Detection. Third, Clinical Performance of a Commercial Multiplex Pathogen Detection System. And an overview of the lessons learned with multiplex nucleic acid recognition method for pathogen detection. In the first part of our program, we will go over the definition of sepsis, the pathophysiology, clinical significance, and the role of early evidence-based treatment on outcomes. Sepsis is defined clinically as the host's systemic inflammatory response to an infectious process, with the presentation of 2 or more signs of a systemic inflammatory response syndrome. The Scher's criteria include elevated or depressed temperature, elevated heart rate and respiratory rate, and elevated or depressed leukocyte count. These criteria are quite general and are not specific to a systemic infection. Expanded signs of sepsis have been developed to build a better clinical picture of the sepsis case. A multifactorial approach is needed to build the clinical sepsis picture. Also note that sepsis can be caused by bacteria, fungi, parasites, and viral infections. The toxins secreted by the organism can cause sepsis. The presence of the organism in the blood is not necessary. For this lecture, we will illustrate the sepsis process with bacteria and fungal pathogens that have disseminated into the bloodstream. Microbiology, a culture-based method, remains the method of choice for aiding in the diagnosis of bloodstream infections. For this presentation, we will focus on bacteria and fungal bloodstream infections. The pathophysiology of sepsis is quite complex. This figure illustrates the interplay between the infectious process, inflammatory response, and the subsequent vascular and hemodynamic changes that results from the release of cytokines and the activation of the coagulation pathway. Coagulopathy in sepsis, such as disseminated intravascular coagulation, can impair tissue and organ perfusion, resulting in organ dysfunction and failure. This lecture focuses on current and novel technologies for accelerating the diagnosis of the infection, in hope that early detection and treatment of the causative pathogen can prevent the progression of the cycle of this disease. So what is the significance of sepsis? About 751,000 cases of sepsis are reported each year in the U.S. Mortality rate ranges from 27 to 54% and as high as 90% with cases of established candidiasis. It is a leading cause of death annually. Now, what is the impact on outcome with early diagnosis and treatment of sepsis? Studies by Kolev reported that inadequate antimicrobial treatment is an important determinant of outcome and mortality. Morrell found that delays in antimicrobial treatment greater than 12 hours was associated with greater hospital mortality in Candida bloodstream infection. With septic shock patients, Morel reported that each hour delay over 6 hours after onset was associated with decreased survival by 8%. Notably, initiation of active targeted antimicrobial therapy in bacteremic pneumococcal pneumonia cases within 4 hours reduced risk of mortality and shortened the length of hospitalization. So the key here is early targeted antimicrobial therapy has a positive impact on reducing mortality and shortening the length of hospitalization. In this next section of this talk, we will go over the current microbiology practice and new molecular tools for pathogen detection. In the clinical setting, if a systemic infectious process is suspected, the physician will order blood cultures to identify the causative pathogen. Urine, wound, aspirates, and other body fluids are cultured as necessary if infection is suspected in those areas. I will use blood culture as an example. For suspected bloodstream infections, at our hospital, 2 sets of blood culture typically are drawn per infection episode. Blood samples are collected into culture bottles using aseptic technique. At our institution, the aseptic process involves the nurse cleaning the draw site with a chloroseptic product. For each set of blood culture order, samples are collected into an aerobic and an anaerobic culture bottle to optimize the recovery of different classes of microbials. 10 milliliters of blood is collected and dispensed into each aerobic and anaerobic bottle for adult patients. For neonates and pediatrics, the volume is 1 milliliter per culture bottle. The larger volume for adult subject is needed to improve chance of recovery of the pathogen in circulation given the larger total blood volume of the patient. With pediatrics or neonates, given their smaller total body of blood volume, they have a greater concentration of organisms per milliliter compared to adults, hence the necessity to collect larger volumes in adults. It is preferable that each set of culture be collected from a different site. The reasoning is to improve the chance of pathogen recovery. There often is a delay in collecting the second set of blood culture. The temporal delay is employed to improve pathogen recovery. This is to account for transient and intermittent bacteremic cases. These photos were taken from our clinical microbiology laboratory. In the upper left photo, panel A, we have a set of blood culture bottles. The blue top bottle on the left is for isolating aerobic pathogens, and the bottle on the right for anaerobes. At the base of the bottle, there is an indicator which signals microbial growth in the bottle by indirectly measuring the CO2 production through changes in the acidity in the media. The indicator changes from green to yellow. Yellow is indication of a positive bottle. The blood culture bottles are placed into an incubator, shown in panel B, and continuously monitored for microbial growth, which takes on average 18 to 24 hours. Once positive, an inoculum of the positive culture is streaked onto growth media plates such as sheep blood agar, chocolate agar, and Brucella. For subculture, and that is shown in panel B, also an inoculum is used for gram staining, and which is shown in panel C. At this point, once gram staining is performed, the morphology as well as the gram stain results reported to the clinician. Speciation and antimicrobial susceptibility is then performed next. Figure A and B shows microbial growth and colonies on subculture plates. Panel A shows sheep blood agar plate with Staphylococcus aureus growth. In panel B, we have a split plate agar On the right half of the plate, we have sheep blood agar, and on the left, a selective media, which in this case is McConkey agar. A McConkey agar often is used for cases with suspected enteric, gram-negative enteric organisms. In this example, E. coli is growing out on the split plate. Laboratorians are able to differentiate lactose versus non-lactose fermenting isolates with the McConkey agar. Subcultures require time to incubate and grow out, which may take another 24 hours. Panel C shows 3 biochemical test plates for organism speciation and antimicrobial susceptibility testing. These plates are used with the BD Phoenix system. The biochemical tests allow laboratorians to profile the metabolic characteristics of the organism, and this is used to speciate. To perform this test, the laboratorian creates what we call a 0.5 McFarland equivalent suspension of the colony in culture broth. The organism suspension is used to fill the test plates. The plates are loaded into the automated reader in panel D, which provides a final result about 16 hours after the plates are loaded into this instrument. As you can see, the culture-based process for pathogen identification is a time-consuming process, requiring about 72 hours for identification and longer for fastidious organisms. Faster diagnostic tests have emerged to accelerate the time to detection and diagnosis of bloodstream infections. These technologies incorporate well-established nucleic acid amplification approaches such as polymerase chain reaction, PCR, for pathogen nucleic acid detection. The gene expert MRSA, methicillin-resistant Staphylococcus aureus and Staphylococcus aureus test by Cepheid is an FDA-approved device. This device uses PCR for its amplification technology. The light cycler Septifast by Roche Diagnostics is a multiplex PCR-based pathogen detection system, which is at present only approved for use in Europe. Both technologies perform thermocycling reactions for amplification of target nucleic acid. I will cover the Septifast test in greater detail in part 3 of my talk. Some other new technology include the linear after the exponential PCR, or late PCR, is a novel derivative of PCR technology that is presently incorporated with Smith's Detection's BioSeq multiplex pathogen detection platform for veterinary and clinical diagnostic application. Other nucleic acid technologies, such as isothermal amplification methods, have emerged. This technology, called LAMP, or loop-mediated isothermal amplification, performs nucleic acid amplification independent of thermocycling, which is an advantage in that it has a potential to be cost savings and has a potential for flexibility for use in multiple settings without the constraints of the laboratory setting, which is often the case with conventional or traditional PCR. Please refer to Dr. Brian Baker's lecture number 5 in our first education course to learn more about the LAMP technology. This is the general workflow of a molecular-based assay. We use the multiplex PCR assay, light cycler Septifast, for whole blood pathogen detection as an example. DNA is extracted from whole blood samples collected in EDTA anticoagulated tubes. Extraction is completed in a biosafety cabinet. The extracted DNA and detection mix, which consists of primers, DNA polymerases, and the fluorescently labeled probes and nucleotides, are loaded into a reaction tube for thermocycling and detection. With real-time PCR, the amount of amplicons, or the amplified target, is monitored at each cycle. Post-processing of the data is by software. With the SeptiFast method, the total analysis time from sample arriving at the lab to reporting a result, is about 6 hours when we are simultaneously batch testing 7 patient samples. This table highlights key limitations of both the blood culture and the nucleic acid recognition methods. Culture methods typically are time-consuming. Growth and recovery of an organism is dependent on the culture media, especially with fastidious organisms. Contamination is always a concern. The key source of contamination often is in the sample collection step, what are called the pre-analytical step. The presence of antimicrobials in the blood sample can reduce the recovery of the organisms by culture. Short sampling, that is, not collecting sufficient sample volume, also can decrease your chance of recovery of the pathogen. On the other hand, with nucleic acid recognition methods, one of the key limitations with nucleic acid recognition is its discrete test menu. The ability to detect the target pathogen is limited by the quality of your primer and probe designs. Contamination also is a concern with nucleic acid testing, as oftentimes these assays are very sensitive. Furthermore, nucleic acid recognition methods detect without discrimination between viable and non-viable pathogens. So nucleic acid testing has the potential to detect remnant or free-floating nucleic acid material released from lysed or dead bacteria. While clinicians may treat pathogens based on the local antimicrobial susceptibility pattern for these isolates, current molecular methods have limited or lack thereof antimicrobial susceptibility testing or data on these pathogens. Some of the current molecular assays for Staph aureus also screen and test for the MEC gauging to determine whether the organism is methicillin resistant, a methicillin resistant strain of Staph aureus. But for the most part, current nucleic acid technologies tests do not offer current commercial assays do not offer resistance gene testing. In this section, I will present an overview of in this next section, I will present an overview of our clinical experience with a commercial multiplex pathogen detection method. We conducted a prospective observational study at UC Davis Medical Center from 2003 to 2006 to clinically evaluate the Lightcycler SeptiFast, a multiplex PCR-based pathogen detection assay. 200 patients were with high clinical risk factor for bloodstream infection and who met the SURE criteria were serially enrolled into the study. They included patients from the intensive care units, general medicine floor, and emergency department. We collected 3 milliliter of whole blood into EDTA tube at the same time as blood culture. Blood cultures were processed by the microbiology laboratory, while the EDTA tube blood sample was used for testing on the SeptiFast assay. The assay workflow has since been refined to work with a lower blood volume of 1.5 milliliter. Please refer to Dr. Nam Tran's lecture in Education Course 1, where he presents his clinical experience with the multiplex assay in a high-risk patient population. That is with the burn patients and the trauma and surgical ICU patient population. These are the pathogens tested for on the SeptiFast assay. The SeptiFast assay is based on multiplex real-time PCR. The target sequence for nucleic acid amplification and detection is the internal transcribed spatial region between the 16S and 23S ribosomal DNA in bacteria and the 18S and 5.8S ribosomal DNA in fungi. Real-time monitoring of amplified products was by hybridization probe technology coupled with fluorescence resonance energy transfer. The quantity of amplified product is related to the fluorescence intensity. This diagram provides a illustration of the principle of PCR and FRET, fluorescence resonance energy transfer. Polymerase chain reaction is an approach for amplifying the gene or the target nucleic acid or the nucleic acid sequence of interest. PCR involves a thermocycling process that consists of 3 steps: denaturation of the DNA, annealing of the primers and probes, and elongation of the DNA strand or the nucleic acid strand by synthesizing the complementary to create complementary DNA strands. At the end of each thermal cycles, we have a doubling of our nucleic acid target from when we started. Typically, PCR assays run between 40 to 45 thermal cycles in a run. Target gene is doubled. The principle of fluorescence resonance energy transfer is the use of fluorescent labeled hybridization probe to monitor for amplified products between each thermal cycle. A pair of labeled probes hybridizes to the target segment. The pair of probes are separated by no more than 1 to 2 bases. During the annealing step, the light cycler emits a specific excitation wavelength on probe number 1. As probe number 1 returns to ground state, the energy is transferred to the adjacent probe, number 2. The wavelength of light emitted from the excited probe 2 is detected by multiple detection channels on the light cycler instrument. We perform thermal cycling reactions with the light cycler 2.0, the instrument shown on the left. On the right is a cross-section showing the components of the thermal cycler. At the top center, we have the thermal cycling core. The carousel shown in the picture on the left at the base of the light cycler is filled with 100 microliter glass capillary reaction tubes, 50 microliter of DNA extract or eluate, and 50 microliter of detection mix is loaded into the capillary tubes. Per patient sample, 3 capillary tubes are utilized, 1 for gram-positive bacteria testing, 1 for gram-negative bacteria, and 1 for fungi testing. The light cycler is equipped with multiple channel detectors, each for a different wavelength. Melting curve analysis is to identify the temperature at which we observe maximum binding of probes. At the end of the thermal cycles for a given run, the temperature of the reaction tube containing the amplified product is lowered and then incrementally heated up while fluorescence is monitored. The maximum temperature differs between organisms, for example, by the different organisms by the ratio of the guanine cytosine rich bonds, and also by the length of the DNA segments. So these 2 factors potentially could affect the melting peak temperature. This is a melting curve profile for gram-negative pathogens on the SeptiFast assay. The multiple detection channel allows for distinction of multiple organisms with similar melting peak profile. For example, Klebsiella in the 640 channel versus Pseudomonas in the 670. The melting peak analysis as displayed on the Lifecycler software, this melting peak profile for the 640 channel is indicative of Klebsiella. This figure illustrates the workflow, the time workflow for pre-analytical, analytical, and post-analytical sample processing on the SeptiFast multiplex assay. The total analysis time, as mentioned earlier, when simultaneous testing of 7 patient samples is about 6 hours. Now the results from our clinical study. The bar graph reports the number of PCR positive and blood culture positive results. The solid bars in black are the raw data, or the raw results. As you can see, PCR detected more positive cases than blood culture. We then screened the PCR and blood culture positive results for contamination, looking specifically at coagulase negative staphylococcus results. Cases with suspected contaminants were submitted to arbitrated case review by a panel of clinicians. And after review by the clinicians, we remove the results that were deemed contaminants, and we observe the final, after removal of the screening of contaminants, 45 PCR positive and 43 blood culture positive results that were deemed and are deemed to be clinically significant. This is the workflow for a clinical arbitrary case review process. The slide shows the arbitrary case review process. The 3 arbitrators are provided with data on the treatment course, laboratory results, patient responsiveness to the treatment, to discern whether the coagulase negative staph results from the PCR or blood culture method are clinically relevant or contamination. This next slide, here are some of the criteria used to evaluate the clinical relevance of the coagulase negative staphylococcus finding. The Venn diagram summarizes the concordance and discrepancy between results from the PCR and blood culture method. The results displayed is the one-to-one comparison between the PCR and the blood culture results or culture samples that were collected at the same time. The large circle on the left represent the number of PCR positive samples, and on the right, number of blood culture positive samples. We observed 19 cases where PCR positive and blood culture positive results were in concordance. They identified the same organisms. We also observed 9 cases involving multiple pathogens detected by the 2 methods with a mixture of concordant and discrepant results. I want to focus and turn your attention and to highlight the 17 PCR positive blood culture negative cases in red. Of these 17 cases, about half of these cases could not be supported. Of these 17 cases, about half of these findings could be supported by others or subsequent culture results. However, there remains several cases which could not be supported by other culture results. These cases were sent to clinical arbitration. The panel of clinicians weighed the results and the clinical case report, and they could not rule out the possibility that the additional PCR findings were that of a real infection. We also conducted analysis to assess the number of patient cases in our study who received inadequate antimicrobial treatment within the first 24 hours following blood sample collection. Here are some definitions on adequate versus inadequate and probable inadequate antimicrobial therapy. Considering the organism detected, and the antimicrobial treatment regimen used, the dosage, and the class of drug used, we found that 25% of our patient population with a PCR blood culture positive result had received inadequate antimicrobial treatment in the first 24 hours following sample collection. We hypothesized that early and rapid identification of the causative pathogen could accelerate early targeted therapy. I now would like to present a couple of cases to illustrate the potential clinical utility of multiplex nucleic acid testing to enhance detection of bacteremia and fungemia and the impact of early detection on patient treatment. This case illustrates early simultaneous diagnosis of multiple pathogen infection. The patient is a 54-year-old male with AIDS and respiratory failure. The patient has end-stage chronic obstructive pulmonary disease requiring tracheotomy and lifelong bi-level positive airway pressure. Patient has a CD4 count of 83 cells per microliter. He was treated earlier in the hospital course for Pseudomonas and staphylococcal pneumonia. He has been on prophylactic therapy with Bactrim for pneumocystic carinae. The patient spiked a fever on the day we enrolled him into the study. At the time, he was receiving highly active antiretroviral therapy. His vitals were temperature elevated at 38.2, blood pressure of 113/77, heart rate elevated 92 beats per minute, respiratory rate of 18 breaths per minute, and the white blood cell count of 9,500 cells per microliter. We subsequently collected the blood culture and a sample for PCR testing. PCR identified Staphylococcus aureus and Pseudomonas aeruginosa. Pseudomonas was not identified by blood culture until 3 days after sample collection. Once Pseudomonas was identified, clinicians added cefepime to the therapy. PCR could have accelerated the identification of Pseudomonas identification and would have prompted the initiation of Pseudomonal infection treatment at least 2 days earlier. Patient received inadequate treatment for Pseudomonas during the wait for blood culture results. This next case illustrates the potential reduction in hospitalization days with earlier targeted treatment. This is a 59-year-old female who presents to the emergency room with lower extremity cellulitis, which progressively worsened over the past few days. Blood culture and PCR was collected on day 1. Patient initially was treated with cefotaxime for her urinary tract infection and cellulitis. She was subsequently discontinued from cefotaxime when her UTI, urinary tract infection, resolved. However, her cellulitis did not resolve. She was then started on hospital day 3 with nafcillin. Her cellulitis resolved on nafcillin treatment. The blood culture collected on admission did not grow out any organisms. PCR isolated Staphylococcus aureus on day 1. The early unique PCR finding could have prompted earlier, possibly 2 days earlier, use of nafcillin, which is the preferred drug of choice for methicillin-sensitive Staphylococcus aureus. The patient was treated primarily for her cellulitis during the hospitalization. So potentially hospitalization days could have been reduced by 2 days with the earlier start of the targeted antimicrobial. To summarize our clinical study and experience with multiplex PCR method for pathogen detection, Multiplex PCR detected potentially significant bacteria and fungi that were not found by blood culture. PCR can serve as an adjunct to current culture methods to facilitate early detection of bloodstream infections, especially in cases where blood culture is negative but the patient is at high risk of sepsis. The PCR method simultaneously detects and speciates microbes faster than culture. Multiplex PCR has the potential to facilitate earlier treatment and impact patient outcome. In the fourth section of our lecture, I would like to talk about and do a recap of the lessons learned from our clinical experience with a multiplex nucleic acid recognition method for pathogen detection. The objectives of this section are to describe and characterize the gaps and technical challenges with current multiplex nucleic acid recognition technology, and to present some integrated solutions to these problems. The gaps and technical challenges. One of the gaps is the discrete test menu with multiplex assays limits the impact of the assay. Resistance gene testing is needed to provide antimicrobial susceptibility data to clinicians. Current multiplex assays lack or do not have antimicrobial susceptibility data or testing or resistance gene testing. As shown in the Venn diagram, with new nucleic acid recognition methods, the lack of standardized comparison method makes it difficult to identify false positives. Blood culture is not a perfect comparison method for nucleic acid testing, which can be more sensitive because the method could be more sensitive than culture. The high contamination rate may relate to the detection threshold of the assay. Here are some solutions to these gaps and technical challenges. As shown in this study, arbitrated case review, as employed actually in our clinical study, arbitrated case review represents a new standard for evaluation of results from new nucleic acid recognition methods. The addition of quantitative testing could help facilitate interpretation of clinical significance of multiplex test cluster results. PCR cycle threshold CT value may allow physicians to discern infection from contamination. Expanding the capability of current multiplex platform to include resistance gene testing will help clinicians profile antimicrobial susceptibility of these organisms. In conclusion, we have the following remarks. Multiplex PCR or multiplex testing can serve in general as an adjunct to current culture methods to facilitate early detection of bloodstream infections. Multiplex testing has the potential to facilitate earlier treatment and impact patient outcome. That is, multiplex nucleic acid testing has the potential to facilitate early treatment and impact patient outcome. We recommend arbitrated case review as the new standard for evaluating results for new nucleic acid recognition methods. And lastly, quantitative testing with new multiplex nucleic acid assays could facilitate the interpretation of real infection versus that of contamination. And this concludes this presentation. Thank you again for your attention.